All right, hello, bio family. So we're going to pick up with chapter one with this lecture video. And the first video that we did in this module, we really looked at the properties of life and the organization of life. And if you remember, when we talked about biology, we said that biology was the scientific study of life or living organisms. So we tackled part of that definition. We define life. Now we're going to go in and we're going to say, okay, what exactly do we mean by scientific study? And like I mentioned in the first video for this module, a lot of you do scientific thinking on a daily basis. You just don't look at it that way. You actually probably go through the scientific method without even knowing it. So we're just going to define what we mean by doing something scientifically and what exactly is the difference between science and then other ways to look at the world. Okay. And so when we look at just the word science, it basically is derived from the Latin verb meaning to know. So basically we are gaining an understanding, we're knowing how something works, how something functions, and it's really based on this idea or based on the process of inquiry. And it really comes from just this curiosity of understanding the world and the way everything works that we're surrounded by. And so biology really then starts from this curiosity and idea of exploration. And so the way it breaks down is that the in biology, we really start with kind of this idea of wanting to know how something works. So we have this initial phase of inquiry where you observe something, you record notes about it, you see something unusual, and you become curious about it. And as you're recording those observations, that's actually data that you're taking down. And data is the foundation or the evidence of scientific inquiry, because that data is what we use to gain an understanding of how something works and how it functions. Um, and so if you think about some examples of data, so just kind of pause the video, think for a minute or two about what exactly are some examples of data. Okay, all right, so let's just kind of give some examples of data. A lot of you probably, when you hear the word data, you initially think about numbers. Okay, so like Excel spreadsheets, that sort of thing. And that is true. That is data. So, for example, if you go out and you measure the height of a human population, that is going to be data. So those actual height statistics, the height numbers. You could do weight. You could do age. You could do all sorts of statistics. Blood pressure, for example. All of those numbers are data. You also could just make observations. You could record and what an organism is doing. So let's say you're watching the behavior of a bird in your backyard. You record everything that bird does, and that's data. Even though it's not numbers, it's data. You're recording that behavior. And so there are different types of data that you can use depending on what your question is and what you're interested in understanding. And so as you work through this process of science, you will generally develop questions about the things that you are observing and watching. And those questions will lead to testing and experimentation. So, for example, let's say you're out in your yard, let's say you like gardening, and you have some flowers. And let's say those flowers have couple of different colors, or those plants have a couple of different colors of flowers. Maybe they have red flowers, maybe they have white flowers and purple flowers. And you just happen to notice that the bees only go to the red and purple flowers. They don't ever really go to the white flowers. And you notice that after several days of watching the bees. Now you become curious, why exactly are the bees not going to the white flowers? So then you can develop a test, an experiment, you can develop hypotheses, and go through the scientific method to potentially determine why those bees do not go to white flowers. Now, in the scientific method, we stated that it's this process of exploration, right? And so that first part again is making those observations. So for example, Jane Goodall, really famous for the observations she made in her research with chimpanzees. She made these observations, she went out, she studied the behavior of these animals, she gathered lots of data, and that led to more questions where she sought information. So here's an example of a page or 2 pages from her notes, where she has a drawing. She writes what the individuals were doing. And again, just because this isn't numbers, this is actually a form of data. And so after making those observations, so like we did with our flowers and our bees, you then will generate a hypothesis. So after that exploration process, you have a hypothesis, okay? And to go to a more simplistic example, and move away from the bee and the flower so we don't complicate it, let's say we have a remote control, and our remote control is not working. Well, it doesn't turn the volume up, can't change channels, so it's not working. So you will generate a hypothesis to your observation of the remote control not working. So a hypothesis is basically a proposed explanation for just a set of observations. And in order for a hypothesis to be valid, it has to be testable and it has to be falsifiable. Those are the 2 big parts of a hypothesis. You have to be able to generate a test, and you have to be able to say whether or not your hypothesis has been proved false or not. So you can have hypotheses be developed in a couple of different ways. Some hypotheses can be developed to where they will have experiments or scientific tests. Or you can have hypotheses that will be developed where you just make further observations. So for example, Jane Goodall probably tested a lot of her hypotheses by continually watching the chimpanzees, where she didn't really, where she probably, I say probably because I don't know a lot about her work in detail, but she probably didn't develop experiments or scientific tests on her chimpanzee community. She mainly answered her questions through observations. But the results of the experiment or observations will either eventually support or not support your hypothesis, okay? And so let's continue with our remote control example. So we have our remote control. We have the observation that when we press any button on the remote, it does not do anything to the TV. Doesn't turn the volume up, doesn't change the channel, you can't pull up the menu, etc. So our hypothesis could be that the batteries are dead. So now that we have our observation, we've generated our hypothesis, now we can make a prediction. And that prediction will be is if we put new batteries in the remote, then it will work. Notice how I did an if-then statement. Predictions come with these, they are these if-then statements. So if you do this, then this will happen. So if I put new batteries in the remote, then it will work the TV. So for this example, the experiment will be to replace the batteries. So once I put the batteries in, If the remote works, then we supported our hypothesis. If we put in our batteries and the remote does not work, then we have no support for our hypothesis. So we will make a different hypothesis, make a different prediction, and then test again. And you can keep going through this process until you generate or until you determine the correct outcome. Not the correct outcome. All outcomes are, that's a bad way to word it. But you can keep generating hypotheses until you figure out why your remote control is not working. Okay. And so that testing involves the revision and drawing conclusions and looking at your results in order to determine a better understanding, or in order to gain a better understanding of why something is functioning or working the way it works in the world. And so the scientific process is this process where you're continually revising and changing, interpreting data, drawing conclusions, so that you can just obtain that better understanding. And so whenever you fine-tune your questions, you really rely on other published data. So you rely on the scientific literature, you rely on published reports, published journals, and you also go to seminars and conferences, and you look at scientific publications to help you gain an understanding in some point to where you may not have had knowledge before. You may be able to go to a conference and say, share your results and talk with other scientists in the field and get suggestions on how you could maybe take your research to really try to gain an understanding of what you're interested in and learning more about. And then also, you know, as we're doing these experiments, before they're published in a scientific journal, they have to undergo this process of peer review where they are evaluated by anonymous. Generally, it's anonymous. Sometimes people will say who they are, but it's generally this anonymous process where experts in your field will review your work. They will look at the quality of your research, your experiments, your data, and they will determine whether or not it's sufficient enough to be published in the scientific community, to be published in the literature. And so that whole process is called the peer review process, and that's really important for kind of checking the work of our community and making sure that we're publishing the best work that we can, and that we're not, you know, really publishing work that shouldn't be out there, basically. And so here we have an example of scientific communication where you have probably some students that are sharing their data, getting feedback on that, on their projects, and that again is helping them with that scientific process. And then after you kind of go through that process, you have a couple of different outcomes that can happen. You can increase your knowledge. You can also help increase the community's knowledge by publishing that research. It could lead to the development of new questions, new research. It could lead to the development of new technologies. It could benefit society. So there are a lot of different ways that the scientific, you know, experiments, scientific method can really benefit, you know, society and the world. And so here's just the summary of basically that process, that scientific process. So you have exploration, you have testing, you have outcomes of that testing, You can communicate those outcomes, and then that could lead to more exploration, or it could also lead to more testing. And so these are, interconnected, interacting parts of the scientific process overall. And so one thing I want to talk about for the next couple of minutes is, we talked about what a hypothesis is. Now I want to talk about what a theory is compared to a hypothesis, because this is something that a lot of the public can get confused about. And I think, in order to be an educated citizen and to be able to evaluate science and, medical information and, all of this other stuff, you really need to know what is the difference between a hypothesis and a theory, because you can hear things like, oh, it's just a theory, doesn't really mean anything, it's just a guess. I've heard a lot of people say that, but actually they have it backwards. Okay, so a theory is actually a really well documented idea in the scientific community. They're very comprehensive. They've been studied. They've been studied a lot. They have a lot of evidence that supports that idea, and an idea, or basically for something to become a theory, it has to have a lot of evidence. It has to be largely supported by that scientific community. And there also has to be an increasing body of evidence leading for support for that theory. And that theory is this general statement that explains observations that we see in the scientific world. And the theory will also lead to new questions and ideas and testable hypotheses that can help increasing our understanding of a specific topic. Theories, however, though, can also be modified. So as new evidence comes up, new ideas, the theories can be refined, they can be changed, and theories can also be thrown out. They can be abandoned if there's really strong contradictory evidence that is found and comes to light. And so for something to be a theory, it is actually really, really well supported, and the idea has a lot of weight. There's a lot to that idea. And if you compare that to a hypothesis is just a guess, an explanation of why something happens the way it does. And so a theory is actually stronger than a hypothesis. Hypothesis probably hasn't been tested yet a lot of the times, because you expect something to work in that way based off of past theory and knowledge, whereas a theory has a lot of evidential support, a lot of evidence supporting that idea. And then when you look in science, there are, we have our hypotheses, we have theories, and then we also can have facts, right? So just to be clear on this, a fact is just a piece of information considered to be true based on the evidence that's presented. So your fact is verified. You can go in and verify it based off of data, based off of evidence. And so it differs and is different from an opinion. And so a lot of the time people will associate facts with science, but science's goal is not accumulating facts. It's being able to explain the happenings of the natural world. And so like we have here, the cornerstone of science is the explanations that apply to the greatest variety of phenomena. Okay. And so those are really the important, components of the scientific method and ideas of science themselves. Okay. And so one final thing I want to talk about here is, experiments and how those experiments work. Whenever you investigate a hypothesis, you oftentimes will perform an experiment to test whether or not your hypothesis is supported or not supported. So there are variables within that experiment that are going to allow you to determine support for your hypothesis. So the variables are the factors that are being changed in an experiment. And you can have a couple of different variables within an experiment. And if you are performing a controlled experiment is where you have a variable of interest and you are only differing one, or you have a variable that you think is causing an outcome that you've observed, and you're only going to change that one variable in a controlled experiment. Okay, so it's where you're comparing two or more groups and only changing one variable between those groups. Okay, and so those groups will be an experimental group and a control group. Okay, so for example, let's say we want to know why. We want to know what affects plant growth the most. Is it sunlight? Is it nutrients? Is it water? The way we could test this is we would control for each of those variables independently. So the first experiment we could do is we could have a control group that, or we would have two groups where we would differ the amount of sunlight that is exposed to those plants. You would have one group that gets, maybe 12 hours of sunlight during the day. And you would have another group maybe gets 6, another group maybe gets 4. But the key thing is you keep all of the nutrients and the water the same between those groups. The only thing you're changing is the amount of sunlight that those groups get. Okay, your control group would be the group that does not receive the specific factor being tested. So the sunlight. So your control group would not get sunlight. And so there it would show, do plants grow with sunlight while they have water and nutrients? And then does the length of the amount of sunlight affect the plant growth? Okay. And so you have your control group with no sun, and then your experimental groups that are varying in that factor being tested. Okay. And so it's important to know how those groups are affected in your experiments and how you have to kind of set up your experiments to get at separating those groups. Okay, so now that we've defined the variables, next we're going to look at kind of the scientific process of scientific method in a real life study. So basically there were some researchers who wanted to determine whether or not baby turtles actually swim, or do they just kind of drift with the currents. And so the background in this is that after emerging from 2-month incubation in the eggs, the turtles hatch, they dig out of the sand, and they use the moonlight coming off the ocean to go to the sea. And so they wanted to know whether or not the juvenile turtles actually swim in the ocean currents, or do they just kind of drift passively in the current themselves. And so what they did is they attached satellite trackers to 24 sea turtles, each of those between the ages of 1 and 2 years old. And what they found is that the turtles move faster and go along tracks that are different from the currents. And they figured this out because they also allowed buckets to drift. And so we know the buckets don't swim. We know they're going to follow the currents. So by controlling And using the buckets, we know exactly what the rate of travel and the direction of travel would be if the turtles only drifted. And so you would expect that if the turtles didn't swim, they would have the same rate of travel and the same direction as the drifting buckets. But the results showed that the turtles actually moved faster and went on different tracks than what the buckets did, suggesting that the turtles swim. And so this is also a graph showing the distance in which the turtles travel compared to the bucket. So the red line is the sea turtles and the blue line are the buckets. So again, evidence supporting the hypothesis that sea turtles swim. And so here we have another type of variable that's being used in this study. So we have what's called independent and dependent variables. So in this experiment, the independent variable is the variable that's being changed, and the variable being changed is the object being tracked. So whether or not it was a sea turtle or a floating bucket. The dependent variable is the variable that you're interested in measuring or determining, and that is the speed of travel or the direction of travel. Okay, so basically in a study, you have an independent variable and a dependent variable. So again, the independent variable is what you're changing, what you're manipulating. So that's the turtles and the buckets. And then the dependent variable is the response or what you're measuring or the outcome of the experiment. So it would be the speed of movement. So how fast are the buckets moving and how fast are the turtles moving? Whenever you're designing experiments, you want to measure one independent variable at a time, or you just want to test one independent variable at a time. So here we're testing turtles and using the buckets to control, and that's what we're changing in that independent variable. So you want to make sure you know the difference between those two. Controlled experiments can also have what's called a placebo, and this is placebos generally in medical terminology, and it's a medical treatment where typically a placebo doesn't know if they're actually getting, say, the effective treatment. And so, for example, if you're testing, let's say, whether or not a medicine affects blood pressure, one group of individuals can get the blood pressure medicine, and then another group of individuals will get a placebo, but they think they're getting the blood pressure medicine. So the placebo could just be like sugar water or a sugar pill. Sorry, not sugar water, but a sugar pill or something like that. And so it's not really the medicine, but they think they're getting the medicine, right? They don't know that they're getting the placebo. And so with controlled experiments, you can have different levels of kind of how you set this up. You can have what's called a double blind, where the participants don't know if they're getting the placebo or the treatment, and then the experimenter doesn't know which group is the placebo. So both of them are blind to who's getting what, and so that means there's no biases within that, right? So if you're giving medication for controlling blood pressure, if a doctor knows that this group is getting the medication and this group is getting the placebo, they may not purposefully, but they may measure the blood pressures differently, not be as accurate in their measurements, that sort of thing. So you want to eliminate those biases. So by the doctors not knowing which group is getting it, they may not, then they're going to measure the blood pressure for each group the same. And then if the people know that they're getting the medication, if they're they know, hey, I'm getting the placebo, they may alter their lifestyles differently. Or if they know they're getting the medication and not the placebo, then again, they could alter the way they undergo their lifestyles, which could affect their results. And so the gold standard for like these medical trials on medications is a double blind controlled study where so for a double blind, the test subjects do not know what they're getting, and the researchers don't know which group is getting the placebo. In a single blind, the test subjects don't know, but the researcher knows. And then if there's a not blind, then everybody knows what they're getting. And so the gold standard is the double blind because it reduces the most amount of biases, and it really minimizes how the data can be affected that way. And so that kind of ends all of the information we're going to go over in terms of scientific study, the scientific hypothesis, how a scientific study set up, the details about the scientific study, and all of those things. And so this pretty much does it for your Chapter 1 module. Next week, we will start our Chapter 2 module, which will focus on chemistry. We'll look at organic molecules, talk about atoms, all of that good stuff. And so we'll see you in Chapter 2.